All right, good morning and welcome to Let's Crack NEET UG English, and we will check the audio first. Okay, I can see few people already there. Saksham, hello Saksham. Okay, let us wait for everyone to join. And please check the audio. If the audio is clear, just message yes so that I know. And I hope whatever we did in the previous class, you have revised that. And in the beginning, like when we start the class, you can immediately post your questions, or you can do it during the session also, so that I can take those points. Sahil, good morning. Asif, hello. Asif, what do you mean by why your videos not in your channel? Actually, I'm taking the classes on all different channels, and I guess everywhere there are videos which I'm putting up, or rather I'm taking live sessions on every channel, right? 3 channels where I'm taking these live on the, say, Let's Crack NEET UG, then Let's Crack NEET, Biology 101. Okay, so I guess the audio part is okay because no one has written anything. And we will continue with our topic, that is photosynthesis. Yesterday was our first lecture and we did some basic things about it. Basic in the sense we talked about the historical background, we talked about the pigments, chlorophyll mainly, and this is what I was talking about earlier also, and I have shared this with you, that I'm taking various chapters on different YouTube channels with Unacademy. One is Let's Crack NEET UG, which is our channel right now, and this is where we are talking about photosynthesis. Because you know, during the sessions the students keep writing that they are targeting NEET 2021 and will they be able to do it. So my answer for everyone is yes, it is possible, and with like if you subscribe to this Unacademy app and take the subscription, the advantage would be that you would be able to get the access to all these various classes on different channels, plus the special classes which I have already started. So this is on Let's Crack NEET UG. Human physiology is what the next I'm starting with, and Biology 101 I've started with human physiology, digestion and absorption. So right now I'm doing 4 chapters on different channels, and 3 channels and 1 on the app. And very soon I'm going to start with the plus class. Those who are new with me, I'm Dr. Neela Bakore, and those who are with me for quite some time now, they already know about it. And this is the app which I'm talking about. If you install this Unacademy Learning app, you get the access to all the special classes. And I've just started with genetics, but very soon I'm going to add a couple of more chapters to it. And once you're on the app, you have live classes of all the educators from physics, chemistry and bio, and live quizzes are there. So it is, analysis also is done on a regular basis. And this is what is the most important thing, because if you decide, or if you are planning for a particular exam like NEET 2021, we know how much time we have. So sooner the better. If you take a subscription, you are like going to get everything what I just mentioned. And these figures, they are going to change very soon, and that's why I want you to take advantage of this. lower price, and if you use my code Neela, then in that case you would get 10% discount. So decide fast and go for the subscription as early as possible. Alright, coming to our topic, that is photosynthesis, and today what we are talking about, in that one factor or one thing that is included is light. Because when we say photosynthesis, in the previous lecture we talked about the requirements: carbon dioxide, water, pigments and light. Carbon dioxide is taken from the atmosphere through stomata. Water is absorbed by the root hair from the soil. Chlorophyll and other pigments are present in the plant itself. Now let us talk about the other factor, that is light. What is essential for photosynthesis? When we say light is essential, photosynthesis takes place in presence of sunlight. So now when we say sunlight, is there a specific wavelength that is essential, or is it like everything whatever is there in the sunlight? So in the solar radiation, what all things are there? This is one thing that we are aware of, which is known as the visible light. The visible spectrum, in which we have VIBGYOR, that is violet, indigo, blue, green, yellow, orange and red. This is our normal white light, which has these 7 wavelengths. So this is the visible spectrum, which is in the solar radiation. Now there is infrared also, then there are radio waves also. If you go other side, it is UV rays, X-rays, gamma rays. What is essential for photosynthesis is the visible spectrum. And that wavelength which is essential for photosynthesis is known as photosynthetically active radiation. PAR is the abbreviation, and on PAR, that abbreviation, also questions have been asked. Now when we say the visible spectrum is required or is essential, so are all the wavelengths which are there in visible spectrum required, or is it different wavelength? Now in the previous lecture we talked about pigments, and pigments are the molecules which absorb various wavelengths. So one particular pigment is probably capable of absorbing say 400 nanometers. The other is absorbing 460, the other is absorbing 600, some other pigment is absorbing 700 nanometers. So every pigment has its capacity or property to absorb a particular wavelength. We have talked about chlorophyll, we have talked about carotenoids, we have talked about phycobilins. These were the 3 categories of pigments that we discussed. Now which pigment absorbs which wavelength? How do we find out? We will have to talk about absorption spectrum and then action spectrum. What is meant by absorption spectrum? We need to find out which pigment absorbs which particular wavelength. For that we will have to isolate the pigments, because if you have all the pigments together, the result is going to be something else, because every pigment is going to absorb according to its property, and you will find that various wavelengths are absorbed by that cluster or collection of the pigments. So we separate chlorophyll A, chlorophyll B, carotene, all these pigments are separated. And then by using spectrophotometer, we try to find out which wavelength is absorbed by a particular pigment. But once we do that action spectrum part, we will realize that the absorption by these pigments is in this range only, that is in the visible range. So visible range, or these wavelengths, this range of wavelengths from this point to this point, is considered as photosynthetically active. That means this is that band where photosynthesis takes place. Or in other words, if a plant is exposed to infrared or ultraviolet, then photosynthesis will not take place, because pigments are not absorbing anything from here. They absorb wavelengths in the visible band. Okay, so now let us come to the absorption part. If we select 3 pigments, here we have selected 3 pigments, chlorophyll A, and we are showing the absorption of chlorophyll A, where it is more, where it is less, by using a black colored line, so that we know which pigment is showing which type of graph. So this black line which is seen here is the absorption of chlorophyll A, and I have written this Vibgyor here. This is that visible wavelength, and when this chlorophyll A is extracted from the leaves, only chlorophyll A, and when we place it in the spectrophotometer, we find out that there are 2 places where the light or that particular wavelength is absorbed. So here is one peak and here is another. So one is in the violet region, which is pretty much in between violet and indigo, and other is in the red region. But normally what happens is, it is considered as this band which is absorbed, because the this particular part is a wide part, this much part. So we say it is right in the blue region and then in the red region. What about chlorophyll B? Very much similar. It is again in this region. So again the band is this much, so it comes closer to that blue again wavelength, and here it is going to be in orange and red. So this is chlorophyll B. Now if we talk about these, chlorophyll A and B predominantly absorbed in violet blue in this part, in this band, violet, indigo blue, and in the red region. So this is, or rather these are the 2 wavelengths where maximum absorption by chlorophyll A and B is seen. There is a slight difference in the peaks which we get, but the band where maximum absorption takes place is pretty much same. So it is blue, violet blue for both chlorophyll A and B, and red again for chlorophyll A and B. So they are absorbing or towards the 2 extremes of Vibgyor. What about this green? This is not absorbed by chlorophyll A or B. See here, both the lines are at the bottom level. This means there is no absorption of this wavelength. Or in other words, we can say if the wavelength is not absorbed, then it is reflected, and that is why chlorophyll A and B, they have this green color. So a pigment absorbs a particular wavelength and reflects a particular wavelength. The ones which are absorbed, they are used for processes, and the one which gets reflected is the color of that pigment which is visible to us. Now what would happen in case of carotenoids? One carotenoid is carotene and the other is xanthophyll. So they are like yellow orange or reddish yellow and orange colored pigments, or little yellowish brown colored pigment. So which wavelength do you think they must be absorbing? If you see it as reddish orange, that means this is the wavelength which is reflected, and that is the wavelength which is received by our eye. So this is not absorbed by them. So they mainly absorb blue and green. So this is, the red line is representing carotenoids. So this band is going to come somewhere here in this part. So it is in the blue-green one. And what is happening in the orange-red, this line is very much on the lower side. That means this orange-red is not absorbed, it is completely reflected, and that is why the pigment is seen as orangish or carrot red, what we call. So when you plot the absorption of various pigments in the visible band, then that is known as absorption spectrum. So it represents the range of wavelengths absorbed by the pigments most efficiently. Absorption spectrum is studied by using spectrophotometer. It is a machine which helps us understand which wavelength is absorbed, and it is very simple. Only time which is required is for extraction of that pigment. So once you extract that pigment, you take that liquid in a test tube and put that test tube into that spectrophotometer, it is going to give you the reading that this is the wavelength which is absorbed most. So that spectrophotometer is used for identifying which wavelength has been absorbed. So this is absorption spectrum. We have represented the range of absorption of various pigments. Now it is very easy. Wherever maximum absorption is done, it is going to be the same place where maximum action is going to take place. Because when we say absorption of a particular wavelength, it is that light or the photons. So suppose in red band or in blue wavelength, photons are absorbed maximum, and these are the photons which are actually going to provide energy for that reaction to take place. So if maximum photons are absorbed in, say, violet blue, so maximum action is going to take place there. If they are absorbed in the red wavelength, then maximum action will also take place there. And what do we mean by action here? So whenever we use the word action in this case, we are talking about the process for which this light is required. And here the process is photosynthesis. There are two ways by which you can find out whether this photosynthesis is taking place or not. One is by finding out the amount of starch synthesized, because in photosynthesis glucose will be synthesized and that glucose gets converted into starch. Or the second method is by finding out the amount of oxygen given out. So there are two possibilities or two different options that we have to find out how much action has actually taken place. Either you find out the amount of starch or you find out how much oxygen has been produced. So what is shown here in action spectrum? It shows maximum photosynthesis takes place in blue and red region. So again, if you are plotting the absorption spectrum, and suppose again I make this VIB-GYOR, this is our visible spectrum or photosynthetically active radiations. We have found that maximum absorption was in this part and then in this part. That is for if you're talking about chlorophyll, okay? So it is just a summed up thing. We are not drawing the separate pigments. And now if we find out how much photosynthesis takes place, we will find that the graph is going to be pretty much same. That means the place where absorption is more, the same place is the one where action is more. So again it is going to be blue and red region. Engelmann, the name of the scientist, measured the rate of photosynthesis as the quantity of oxygen released. We have talked about two options. Either we find out the amount of product, that is carbohydrate, or we find out the amount of byproduct, that is oxygen. The way Engelmann calculated it was this method, that is how much of oxygen is given out, that is byproduct is given out by green algae when they were exposed to various wavelengths. You remember we talked about some basic experiment which was done. There was a long tub taken, green alga cladophora was placed in, and VIBGYOR, various wavelengths were provided. Because of this long tub, the wavelength was focused only in one region, and it was found out that in blue region and in red region there were maximum oxygen bubbles which were produced. And why would this oxygen be produced? It is a byproduct of a particular process. What is that process? The process is photosynthesis. So this is how we are able to find out in which wavelength what maximum rate of photosynthesis is seen. The experiment showed that green algae release maximum oxygen in blue and red region. Oxygen is a byproduct of photosynthesis, that is more the byproduct means more the action. This is how we are going to write things. See, one is the understanding part. Once you understand it, it's perfect. But if you have to write the answer, like the students who have to appear in 11th class also, basically, you know, when we are studying, we are actually targeting either one exam or two exams. The 11th class students, they are targeting two exams. They have to keep in mind the 11th class exam as well as we have to prepare for NEET. In 11th class, the exam is subjective pattern, and in NEET it is MCQs. So if it is multiple choice question, the concept has to be in our head. We don't have to write anything. But in 11th class or 12th class exams, subjective answers are to be written. So unless and until you write everything in a systematic manner, you don't get the marks there. And for competitive exam, that is NEET, we need to understand the concept. Nobody is going to ask us to write anything. So the interpretation of our concept or understanding is checked in the form of multiple choice question. Okay, so that is why my focus is explaining things, and I'm also giving you the method in which you need to write things down, so that you're preparing for both your exams simultaneously. And if we have like the students who have taken a drop, then that is fine, because we don't have to write anything, we have to just understand the concept. So absorption spectrum and action spectrum, clear? This statement also says the same. Action spectrum parallels the absorption spectrum. It indicates that photosynthate form is related with light absorption. And you have to remember the name of the scientist, that is Engelmann. So this is about our action spectrum and absorption spectrum. And now we know what is meant by PAR, photosynthetically active radiation. So far, whatever we have done, is there any doubt, any query related to action absorption spectrum? Ajay, did you join little late? Hello? Okay, so I'm just giving you a few seconds. If there is any doubt, you can message me on action and absorption spectrum, because now after this action absorption, we are going to start with the fluorescence part of chlorophyll, and then we'll come to the photosystems. Okay, so far no message, that means everything is clear, and you can still compile your question and message me towards the end of the lesson. What is meant by fluorescent? I'm sure you've heard these two words, fluorescence and phosphorescence. Fluorescent dye, fluorescent colored t-shirts, the highlighters that you use, they also are fluorescent. Suppose you compare yellow, a normal yellow color, and a fluorescent yellow color. The fluorescent yellow is little more bright. It can be easily detected. I'm sure you've seen t-shirts on which some text is written in fluorescent dye. This is one thing, okay? It glows. On the normal yellow, rather there is a difference between the normal yellow and the fluorescent yellow. There is a difference between normal orange and fluorescent orange in every color. This is one part. The other is phosphorescence. I'm sure you've heard of that when there are children, small children, in their rooms, normally what parents do is they put some stickers, maybe stars, maybe moon, anything, you know, there can be Barbie stickers which are placed on the roof of the room. And what happens is, when the small children, they are made to sleep and the lights are switched off, all those stars and moons and everything, they glow. That is phosphorescence. So let us understand these two things, and we will see that out of the two processes, fluorescence and phosphorescence, fluorescence is also exhibited by chlorophyll. Suppose this is the minimum level of energy which the electrons of a particular pigment is in. This is a pigment molecule, a pigment molecule, and in that molecule one atom has this electron, and this electron is in its normal energy level. Now when light falls on it, the pigment absorbs photons, and that energy excites this electron. So the electron from the lower energy level now reaches the higher energy level. So the electron comes here. Why was the electron able to come here? Because of that energy which was available. So sunlight, the visible spectrum, is the one which is providing this energy. So electron takes that energy in the form of photons, and from lower energy level it jumps to higher energy level. Every molecule tends to come back to its original energy level. So now what will happen is, this electron will tend to come back to its normal energy level. From lower it went to higher because energy was given to it in the form of photons. From higher it will come down to its normal energy level, and from higher, when it is coming to lower, all that extra energy will be given out in the form of a wavelength, and this provides that extra brightness to that pigment. So if you have yellow, fluorescent yellow, what is that pigment doing? When light falls on that fluorescent yellow pigment, the electrons get excited. When those electrons from higher to higher level come back to the normal or lower level, they emit that extra energy in the form of visible light. So that yellow appears extra bright. This is fluorescence. What happens in phosphorescence? This is fluorescence. Now let us talk about phosphorescence. Pretty much same thing with a slight difference. This is the electron at lower energy level. Okay? Now when sunlight falls on this, or when the photons are available for this electron, what will this electron do? This electron will get excited and will come to the higher energy level. Now, between the higher energy level and the lower energy level, there is a transition band. This electron, it comes back and gets trapped in this transition band. So what is happening? When it is coming down, it releases energy, then it goes back, then again comes down, releases energy, again goes back, again comes down, releases energy, again goes back. So now, as long as the source of light is available, fluorescence will continue. Now, in case of phosphorescence, suppose we remove this source of light. So, electron got excited, it gets into transition and remains there for some time. So, even if the source of light is cut off, because the electron is moving in the transition band, and whenever it comes down in the transition band, it releases that energy. And when it releases that energy, it will be seen in the form of a glow even after the source of light is cut off. This is called phosphorescence. So the stars and moon and constellations, everything which is put on the ceiling in the form of stickers, what are the stickers made up of? They have something, some molecule which shows phosphorescence. But what is written on our t-shirt is a fluorescent ink, or what is used to write on the t-shirt is a fluorescent ink. So when you are in light, that fluorescent text is very brightly visible. Does it glow when you go in dark? This process stops as soon as the source of light is cut off. Okay, so difference clear between the two processes? Chlorophyll shows this process, fluorescence. So now what happens in chlorophyll? There are chlorophyll molecules, and now we know the structure of chlorophyll. It has magnesium, nitrogen, carbon, so many things. From the atom, suppose when light falls on this, what will happen is electron will get excited, and when the electron gets excited, from lower energy level, those electrons will come to higher energy level. So this electron, normally a pair, it comes here at higher energy level. And these electrons will come back to the original. And when they come back to original state, some energy is lost in the form of light. This is possible if you extract chlorophyll. So these molecules which we have drawn are extracted chlorophyll molecule. So it is only extracted molecule which is going to show this process of fluorescence. This process is not seen when the chlorophyll molecule is in the system, that is in the chloroplast or in the leaf, because there the process is same, but the plant is not going to waste this energy in the form of light. Plants are going to use this energy which is lost here to generate ATP. So if you extract chlorophyll molecule in a test tube, you will see this phenomena of fluorescence. But same thing when happens inside the plant system is efficient to capture this lost energy to generate ATP. So energy is solar here, and then this is chemical. So this is what is done. The first step in photosynthesis, that solar energy is captured by chlorophyll and is used to generate chemical energy. So plants have the capacity to convert solar energy into chemical energy. So this fluorescence part is clear. It is shown by chlorophyll molecule, but the chlorophyll molecule has to be extracted. If it is out of the system in a test tube, then only this phenomena is visible. Now let us come to this photosystem, which is the most important thing, and this is where this fluorescence thing will be used up. So what exactly is a photosystem? Photosystem has many things. We will draw photosystems, then we'll come to the types of photosystems. So suppose there is, and I'm using green for chlorophyll. This is one chlorophyll molecule, and then we will make some other chlorophyll molecules also with a little lighter green. And along with chlorophyll molecule, there are some others also, like the carotenoids, xanthophylls, all those. So there is a collection of pigments. We have to label this because we are doing it for the first time, and after we have understood this, then we will not label it. This is called the reaction center chlorophyll, and reaction center chlorophyll is always chlorophyll A, no exception. So our reaction center chlorophyll is going to be always chlorophyll A. The other pigments that we have drawn, they are chlorophylls, carotenoids, xanthophylls and all. They are all called accessory pigments. Accessory means helping. Now when sunlight falls on this collection of pigments, suppose this is the source of light and sunlight falls on all these pigments. Now depending upon the capacity of a particular pigment, the pigment absorbs photons, whatever the capacity is. So suppose this pigment has absorbed some photons, it is going to pass on those photons to the next molecule. From here the photons will be passed on to reaction center chlorophyll. Same thing is going to happen with this molecule. It absorbs some photons, passes on to the next molecule, this passes on to the next molecule, and ultimately all the photons are passed on to the reaction center chlorophyll. So here pigments absorb photons and pass them to reaction center chlorophyll. So ultimately what is what is the job of these accessory pigments? The accessory pigments are going to absorb photons according to their capacity, and that is why we are saying that they are helping in this process. Once the main chlorophyll, that is reaction center chlorophyll, gets enough photons, the electrons which are present in that reaction center chlorophyll, they get excited. For our understanding, we are drawing a molecule, but we will draw that molecule slightly away from it. See, it is all inside a membrane. Chlorophyll is present in the thylakoid membrane. So all these are like pretty much very close to each other. So if this is reaction center chlorophyll, there is a molecule which is going to immediately take that excited electron. But I'm going to draw that molecule little away from it. Okay? And this is called the primary electron acceptor. So as soon as the electron from reaction center chlorophyll gets excited, and it is always a pair which gets excited, so this pair of electron, it goes from lower energy level to higher energy level, and it is trapped by the primary electron acceptor. And this complete system is known as a photosystem. What is the job of these pigments? Reaction center chlorophyll is the one from where the reaction, that is photosynthesis, is going to start, because the electron is getting excited from that chlorophyll. Other pigments, their job is to capture, collect light. And when we collect something, we use a word which is called harvesting. So this collection of pigments is known as light harvesting complex. Same collection can be given one more name. Right now also we see those, but in earlier times, like I'm talking about 10-15 years back, there used to be antennas. So if you had a television, you would have an antenna on the roof somewhere, and the antenna was rotated in such a manner that it could gather the particular frequency. Similarly, this pigment collection is collecting the photons. So it is also known as antenna complex. So what all things are there in our photosystem? Our photosystem has reaction center chlorophyll, light harvesting complex, and its primary electron acceptor. So a photosystem has reaction center chlorophyll, which is always chlorophyll A, plus it has light harvesting complex. These abbreviations are used, okay? Light harvesting complex, and of that complex, the primary electron acceptor. These three things would make the photosystem. Now we have understood what is the photosystem going to do like. When light falls on these pigments, they will capture photons according to their own capacity, pass it on to reaction center chlorophyll. Electron will get excited and it will be accepted by a molecule that is called primary electron acceptor. See, very similar to what happened in fluorescence. Now, if it was the outside living system, that is, if we are doing it in a test tube, then that electron will come back to its level and energy will be released in the form of light. Here, that excited electron has been captured by primary electron acceptor, and now this electron will be passed through a chain, and that chain is called electron transport chain. And the energy released when the electron comes from higher to lower level, that energy will be used to generate ATP. Now, reaction center chlorophyll, it is also a pigment, so that chlorophyll will also absorb wavelength. So chlorophyll A can absorb two types of wavelengths, or if I have to put it in a different manner, say reaction center chlorophyll. Now if this reaction center chlorophyll A absorbs 700 nanometers, and one chlorophyll A, it is also reaction center chlorophyll, it absorbs 680 nanometers. So there are two types of chlorophyll A molecules. Both are going to be reaction center chlorophylls, but their capacity is different. One absorbs 680 nanometers, the other absorbs 700 nanometers. So if in our photosystem our reaction center chlorophyll absorbs 700 nanometers, we will call it photosystem 1. Okay, so what is in photosystem 1? In photosystem 1, there is chlorophyll A. It is written as P, pigment, and 700, plus light harvesting complex, plus primary electron acceptor. This is PS1, photosystem 1. And if our reaction center chlorophyll absorbs 680 nanometers, then it will be called PS2. So what is in this PS2? PS2 has chlorophyll A, which is a pigment, and it is written as P680, plus light harvesting complex, plus primary electron acceptor. So there are two photosystems. Both the photosystems have same things. Both have reaction center chlorophyll, both have light harvesting, both have primary electron acceptor. Only difference is in PS1, the reaction center chlorophyll A absorbs 700 nanometers, and in PS2, the reaction center chlorophyll absorbs 680 nanometers. Now, why this 1 and 2 number? This is on the basis of discovery. So, to PS1 and PS2, 1 and 2 numbers are given on the basis of discovery. So, the one which was discovered first is termed as PS1. The one which was discovered later on is called PS2. So, is this photosystem part clear? So, if this photosystem part is clear, then it's okay. If you have any doubt, then let me know, because this is the starting point for our light reaction. Okay, so let me check if anybody is messaging anything. Supayan Mukherjee says, ma'am, will you complete entire syllabus? Yes, this is what I'm trying, and that is why I'm always, like in every lecture, I'm telling you that, you know, take the subscription, join the special classes, join the plus classes, because very soon I'm going to be taking multiple chapters on these special classes and plus classes. But for that, you need to decide fast and take the subscription. Ayush says, clear, everything is clear. Ayush says, now I have understood why it is called antenna complex and light harvesting. Okay, so that is the purpose of me taking a class, because in the beginning I said that I want you to understand it. Up till now, probably you were just mugging up that this is called antenna, this is called antenna, this is called light harvesting. But now when you know that why it is called so, you don't have to revisit those things. And this is what is biology. Understanding is the most important thing. Now tell me, Ayush, do you have to go back to this and say that, you know, this is called antenna, this is called antenna, or once you have understood, now I don't think you will have to think about it second time. What happens to infrared? They're there. The plant is not going to use it. Plants have pigments, and pigments have the capacity to absorb the wavelengths which are in that visible band. Okay. So all other wavelengths, they are photosynthetically inactive, because that visible one we call PAR, photosynthetically active, and others, because they are not helping in this process, they will be photosynthetically inactive. Supayan says, crystal clear. I am so happy to know that these concepts are becoming clear. And there is going to be a check for me also once we have a quiz after completion of this chapter. And then if you're able to answer all those questions, then only I'm going to believe that all these concepts are absolutely clear to you. Okay, so far so good. So now we know that there are two photosystems, and the difference between these two photosystems is only one, and that is of which type of chlorophyll A is acting as a reaction center, whether it is 700 nanometer absorbing molecule or 680. So this is the basic. Now next time when we draw non-cyclic photophosphorylation and cyclic photophosphorylation, we are not going to talk about all these things in detail, okay? So before the next lecture, this part has to be revised. Emerson's effect and Emerson's red drop. Emerson, the name of the scientist, what exactly was done by him? What he did was, he took plant, exposed this plant to 700 nanometers, and he found that there was some rate of photosynthesis. Say that was X. This is just for our understanding. Then he took the same plant, exposed it to 680 nanometers or lower wavelength. One was a higher wavelength, one was lower wavelength. And he measured the rate of photosynthesis. That came to Y. So when the plants were exposed to one wavelength, some rate of photosynthesis took place. And how was the photosynthesis calculated? By the amount of oxygen which is released. Now he took the same plant and gave both the wavelengths simultaneously, and then calculated the rate of photosynthesis. It was more than X, more than Y. It was more than X and Y. What does this indicate? It indicates that there is a system in the plant which works at a particular wavelength, say 700. But there is one more system in the plant which is working at 680 nanometers. And that is why when you give both the wavelengths, both the systems are working and the rate of photosynthesis is more than X and more than Y. So this led to the conclusion that there are two photosystems. This is one part. Then, now when light was given, various wavelengths, so some photosynthesis took place in this part, some photosynthesis took place in this part, and after red wavelength there was a sudden drop because the wavelength visible band stops here, and after red there is infrared which is photosynthetically inactive. So after red wavelength, sudden drop in the rate of photosynthesis, and this, because it is taking after the red wavelength, it is called red drop. And don't you think here the rate of photosynthesis is more? So can we call it enhancement? If, again I'll go over this, if only 700 nanometers wavelength was provided, some photosynthesis did take place. Then when this wavelength was provided, again some photosynthesis took place. When both the wavelengths were provided simultaneously, the rate of photosynthesis was higher. This was called enhancement in photosynthesis, and it was given by Emerson. So it was called Emerson's enhancement effect, or it is also known as simple Emerson effect. Okay, so okay, Ankit says, ma'am, I'm not comfortable with English explanation. Ankit, there is another channel that is called Let's Crack NEET UG, where it is mixed Hindi English. There is one more YouTube channel, it's called Biology 101, where it is again mixed. There is a special class which I'm taking, but for that you need the app. And on the special class also it is English Hindi mix. It is the only channel where we are right now, that is Let's Crack NEET UG English, which is pure English. And that is why I'm giving you that schedule in the beginning of every session, so that you can decide which channel to watch. So except for this one, on all of the platforms I'm taking Hindi English mix, so you can come to the special class also. Okay, why we need two photosystems, ma'am? Actually, we don't need, they are present there. And more the number of photosystems, better the efficiency, because you have a band of wavelengths. And if the plant had only one photosystem, only one wavelength would have been captured. So to increase the efficiency, the plants have two photosystems so that they are able to capture more and more of that wavelength. Okay, so now is Emerson's enhancement effect and the red drop parts clear? So there are two photosystems, that was also told to us by Emerson. There is a sudden drop in the rate of photosynthesis after red wavelength, that was also given by Emerson, and it is known as red drop, Emerson's red drop or only red drop. And when both the wavelengths are given simultaneously, there is an increased rate of photosynthesis, and that is called Emerson's enhancement effect. Now, what is the site of photosynthesis? Site is the place where photosynthesis takes place. The site is chloroplast, and I'm sure we know about chloroplast. Chloroplast is a double membraned organelle. It is a type of plastid. The inner membrane is smooth, not like mitochondria. In mitochondria, the inner membrane is thrown into folds. Here, from this inner membrane, there are some infolds, and from the infolds, a small piece is going to pinch off. And a new piece comes, so one or older one gets pushed, one more comes, the other gets pushed, and so on. So there is a stack of those smaller pieces. So you would see such kind of stacks. But all the time you will find that these stacks are somehow connected to the membrane. And here is a jelly-like material. This jelly-like material is called the stroma. This complete stack is known as a granum. Singular is granum, but if you're talking about plural, then they are called grana. Okay? Plastids or chloroplast, if you're talking about specifically, is a semi-autonomous organelle. It has its own DNA, and it is semi-autonomous because it has its own DNA and it can synthesize its own proteins. And for protein synthesis, it has ribosomes also. And these ribosomes are 70S type of ribosomes. This I'm sure you've studied in the chapter of cell structure and function. The stroma has enzymes for dark reaction. Because enzymes are present here, stroma is the site for dark reaction. Now, where is light reaction going to take place? Wherever the pigments are. So, if this one piece that we are talking of is called thylakoid. So, if we enlarge a thylakoid, suppose this is one thylakoid that we have enlarged. So, this is going to be the membrane of thylakoid. This is the membrane of thylakoid, and the inner space is called the lumen of thylakoid. Why are these things important? Because in next lecture we will be talking about photophosphorylation. That's where this is going to be very important, and then when we come to chemiosmotic theory, this is going to be the most important thing. Where are the pigments? The pigments are in the membrane of thylakoid here. So this is where we have pigments. So wherever the pigments are, that is the place where the light will be absorbed, and the place where light is absorbed, that is the place where light reaction is going to take place. So grana has thylakoids, and this is the site for light reaction. So when we talk of chloroplast as the site of photosynthesis, we need to know where exactly light reaction takes place and where exactly dark reaction. So stroma is the site for dark reaction, and grana or thylakoids are the sites for light reaction. So this is for light reaction, and this is the site for dark reaction. There are two types of chloroplasts. So chloroplasts are dimorphic. Again, I assume that you have studied this in the chapter of cell structure and function. If not, then this is one thing which we would need again when we come to C3 and C4 cycle. So we will take that up when we come to C3 and C4 cycle. But whatever we have done so far, the photosystem part has to be revised. The site of photosynthesis has to be revised before our next lecture, that is tomorrow. So I will see you tomorrow at the same time. Don't forget to revise everything, and after you revise, I'm sure you are going to find some doubts. So when we meet next, we will start with your doubts in case if you have some. Alright, so till tomorrow, take care, and I'll see you there. Bye bye.